But we don't really care about that, right? No. We care about the antimicrobial effect of sebum. Now, sebum is really cool because it lowers the pH of your skin, which makes it very hospitable to some bacteria. So you lower the pH of your skin, and most bacteria, if you talk about pH plates, right, most bacteria don't like low pH, so they won't be able to grow on your skin because of the sebum. Got it? Now, of course, some bacteria they bypass that. Okay, we know, for example, Staph aureus likes salt. So that's why you can have Staph aureus on your skin, even though your skin tends to be a little salty. So Staph aureus doesn't mind high salt. Staph aureus doesn't mind low pH. It hangs out just fine. Okay. Now let's talk about the inside skin, the mucous membrane. And as we talk about the mucous membrane, keep in mind what are the differences between the mucous membrane and your outside skin, basically the epidermis, all right? Now the mucous membrane, it tends to be on the thinner side. So I said the epidermis is thick, right? Thick layer. The mucous membrane, the inside skin, coated with mucus tends to be a little thinner. The cells on the surface of the mucous membranes, they are alive. Compared to the epidermis, right, they are dead. Now, these next two lines here help to tell us how the mucous membrane protects us from bacteria and other pathogens. Now, compared to the skin, you're like, well, what can the mucous membrane do? It's already on the inside of you, right? If you've got bacteria in the mucous membrane, they're in your body, makes sense? Well, how do we get them out? Well, I did say this mucous membrane is thin, but those cells, just like in the epidermis, they are packed tightly. So they may be thin, right? But they're packed close together. So it helps to prevent bacteria from getting entry into your blood vessels. Got it? Also, just like with the epidermis, we have a continual shedding of the outermost layer of the mucous membrane. Even though the outermost layer, they're living cells, we are continually sloughing them off. And if there's any bacteria on that outermost layer, and if you slough it off, right, they're as good as gone. Makes sense? No. I won't focus on that last part. Why? Because it really doesn't help us in terms of preventing bacteria, preventing viruses, preventing worms from getting entry into your body. Okay? Now, this slide I want to focus on what can we really do in terms of what's an effective way. So what is an effective way for the mucous membranes to get pathogens out? Okay? So let's focus on the trachea here. Focus on the trachea. So some cells in the trachea, they produce a very thick and sticky mucus. So if you've got any pathogen within that trachea, they're going to get stuck in that thick mucus. Likewise, along the trachea, you have cells lined with cilia. So think of cilia like little hairs, right? And they move. And their job is to get rid of any mucus, right? So they're basically going in reverse, right? Anything stuck in the trachea, like bacteria, they're stuck in the mucus. And the ciliated cells do this, right? They get it out of the trachea. Now, you either cough it up, right? Or you swallow it. Either way, it's out of the trachea. So basically, nothing gets into your lungs where it can cause severe disease. Make sense? Okie doke. Table 15.1 does a nice job of comparing and contrasting the jobs of the skin, epidermis, versus the inside skin, mucous membrane. We've already talked about number of layers, how tightly they're packed, dead or alive, mucus present, moisture, right? Defenses, again, I'll talk more about them later. Lysozyme, sebum, cilia, and constant replacement. That's a nice job of summarizing everything I just said. All right. Let's talk about your tears. Again, we're all talking about what? First line of defense, that's your barrier. Keep the pathogens out, okay? Now, of course, your tears, made by the lacrimal apparatus. I'm not going to quiz you on that. Focus on what the tears do, okay? Of course, when your eyes become irritated, you make lots of tears, correct? The tears basically wash your eyes of the irritant, okay? Also, tears have lysozyme. I mentioned that earlier, correct? Lysozyme, the same that's on your skin, breaks down the cell wall of bacteria. That's a good thing. Also, just like your sweat, tears are what? Salty, right? So tears have salt. That salt is still inhibitory, just like the salt on your skin. So tears have salt. Bacteria don't like that salt, right? Draws water out, okay? Tears have lysozyme, puncture holes in the cell wall, and also mechanical, right? Just flush the irritant out, okay? So that's how tears help you from getting sick. All right, we'll go into all of that anatomy. It's not A&P 2 or 1, okay? Now, let's talk about our normal flora. We can't count them out, right? We were talking about them for the past couple of chapters, correct? In terms of how they help prevent pathogens from taking root somewhere, okay? That whole concept of your normal flora competing with potential pathogens, that's called microbial antagonism, okay? So they are competing for the same space, right? They're competing for the same food. Okay? That's microbial antagonism. That helps you, okay? That helps you. Now, what are the normal flora? I like to say same word, right? Microbiota, normal flora. Why are they making it hard for pathogens? Okay, first of all, they are consuming the nutrients, right? Right. No food left over for the pathogens. They also create an environment that's not suitable for your pathogens. So sometimes, for example, your normal flora makes or creates a certain pH in their own environment. Now, the pH they create in their environment within your body is good for them, right? Not good for a pathogen from the outside trying to take up residence in you. Okay, that's a good thing. All right. Also, your normal flora actually helps your body to stimulate an immune response. So it's been shown that animals grown in a sterile or azenic environment, right, which means they have no normal flora, if they're exposed to pathogens, they take a long time to respond to those pathogens. So your normal flora helps you to respond quickly to any pathogens that are trying to invade you. And lastly, your normal flora helps to keep you in good health. If you're healthy, right, you do a good job in defending the body against what pathogens versus when you're sick, makes sense? So I've already mentioned before that E. coli in your gut makes vitamin K, right? You need that. Some other bacteria make some vitamin B. That's not essential because you can get vitamin B from your diet, makes sense? Vitamin K is really essential because you can't get it from your diet, you can't buy it either. But essentially, your normal flora in the gut, they make vitamins for you and it helps keep you in good health. Got it? Now let's get back to our defenses. I promise you I'll talk about it. Well, here it is, right? So these are basically little antibiotics, essentially. They're short proteins present on the skin, along the mucous membranes, and also neutrophils have them. Oh gosh, that's a new word, okay? Neutrophils, I'll give you a real quick entry. I'll talk more about them, more than you ever care about later. Neutrophils are basically types of leukocytes, basically types of white blood cells. White blood cells, they are your soldiers in the army, right? They help you to fight bacteria, viruses, worms, etc. They act against a variety of microbes. You can think of them as a broad spectrum antibiotic. They work against gram-positives as well as gram-negatives. They work in several ways. For example, they too can puncture holes in cell membranes. They can also inactivate enzymes in bacteria. So if you take out enzymes in bacteria, they can't do certain things, correct? If they can't do certain things, they can't live, correct? Now, other processes and chemicals, they can help you too. So organs secrete chemicals with antimicrobial properties. I've already mentioned the stomach, correct? Last time I checked, stomach has stomach acid, right? Right. Low pH, okay? Most bacteria don't like that, okay? Of course, there are exceptions like what? Helicobacter pylori, okay? It can withstand that. It actually makes enzymes to neutralize the acid. That's why it can hang out in the stomach, make ulcers, but most bacteria can't do that. So if they get into your stomach, they're as good as toast, makes sense? They're dead. Also, think about saliva in your mouth. Now, yes, saliva has lysozyme, but also saliva actually washes or bathes your teeth from bacteria. You're like, well, why does that even matter, okay? People with dry mouth, okay, so some diseases, some syndromes, you actually do not produce enough saliva, you have a dry mouth. They are more susceptible to dental caries, makes sense? In other words, they have more bacteria in their mouth because they don't have saliva to get rid of them. So they're more likely to have tooth decay versus someone else who has a normal amount of saliva. Makes sense? Okay. All right. Here is just talking about different things in your body in terms of how they work. I talked about saliva, talked about the stomach somewhere here, stomach acid. All the other ones, it's nice to know, but I won't test you on that. But I did mention saliva, and I did mention your stomach. Okay. All right. Oh boy. So what if this pathogen is very, very sneaky, right? it got past your first line of defense. Got past the skin, right? Got past your mucous membrane, got past your tears. Now what, right? You go to plan B. This is your second line of defense. Now compared to the first line of defense, the second line has no barriers, okay? No physical barriers in your second line of defense. Mostly you're talking about cells like phagocytes and other antimicrobial chemicals we'll talk about, like interferons and complement proteins. Right? Now, many of these components, whether they're proteins or cells, they originate from the blood. So that's why in your second line of defense, there are no physical boundaries here, not like a skin or mucous membrane. Now, in terms of the blood, we got to talk about the blood because many of these things come from the blood itself. Your second line of defense, okay? Of course, the liquid part mostly is plasma. Yeah. Let's get to the good part, right? Here we go. Within your blood plasma, you actually have compounds that bind and transfer iron. You're thinking, why is this important? Every living thing needs iron. Iron is a very important cofactor for many enzymes. If you're talking about electron transport chains, many of those cytochrome proteins have iron in them, okay? If you're talking about your red blood cells, the only way your red blood cells can carry oxygen is by hemoglobin, which has iron. So if you're living, you need iron. Makes sense? Okay. Now, in your body, for example, you have little proteins called transferrin and lactoferrin. Their job, basically transfer iron within the body. You basically grab iron, right, hold on to it, and transfer iron throughout the body. That's their job. You don't want free iron floating around because any free iron will be available for any pathogen. You don't want them getting it because they'll flourish, okay? Hit it.
Would they be able to digest the iron since wouldn't it be in the Fe+3 state instead of the Fe+2 state? Because that happens in your stomach. So how would they be able to utilize the iron floating around in your body?
They steal it.
But so can they? So they can ingest Fe3. Okay, I didn't know that.
Yeah. So whatever you're carrying, they steal it from you, literally.
Okay.
They steal it. They steal it from you. Okay, does. Now these two guys, let's focus on transfer. Transfer basically transfer iron throughout your body. Okay. Now, some pathogens, like our favorite Staph aureus, is very, very sly, okay? It wants iron too, okay? So Staph aureus, for example, it can make siderophores. So siderophores are made by Staph aureus. And siderophores basically steal iron away from transfer. Literally. Because Staph wants iron too, okay? It's like a tug of war. Basically, siderophore has a higher affinity for iron compared to transferrin. When that happens, your body responds to that. So now your body makes lactoferrin in response to staph trying to steal your iron. So lactoferrin has a higher affinity for iron compared to the siderophore. So if you can get more lactoferrin going, you win the tug of war. It's like who gets the iron? You're both fighting for it.
So first step, transferrin and siderophore are fighting, and then transferrin calls in some troops. Yeah, say, hey, come on over, guys.
Exactly. So he's the primary guy transferring iron. If this guy's there, he's going to steal iron from him. He's going to say, I need backup. Now your body makes that one. This guy steals the iron away from that guy.
Good.
It's a tug of war. But staph, okay, is very keen. Even in that tug of war, staph will say, okay, I'm not going to fight that fight anymore. Well, staph makes an extracellular enzyme called hemolysin.
Mm-hmm.
And we talked about blood agar plates and hemolysis, okay, you got to make the enzyme to do that, right? Hemolysin. Now, what is staph trying to do? It's trying to break open what?
Blood cells.
Red blood cells to steal your iron.
Wow.
So that's why you see those patterns on blood agar. If they can lyse the blood cells, they're trying to steal the iron away. They go past all of that war, right? They just go right to the red blood cells, lyse it open, grab the iron, and they just multiply. Okay. That's cool. Complement proteins we'll mention today. Antibodies, of course, that's what? third line of defense, that's chapter 16, after your test on Tuesday, okay? But we will talk about complement proteins today. Basically, complement proteins complement the immune system, okay? All right. Now let's talk about the actual cells within the blood. They're called formed elements. You've got 3 types of formed elements, basically cells in the blood, okay? Erythrocytes, sure, carry oxygen, carbon dioxide, we don't care, right? It's not A and B. Platelets, sure. Blood clotting, yeah, we don't care, right? Not in this course. We care about the last one, right? Leukocytes, these are your white blood cells. These are your soldiers in the war, okay? They basically defend the body against invaders. That's their job. They're your soldiers. They're divided into 2 groups, granulocytes or agranulocytes. Now, A, whenever A is in front of any word, it means what? Without something, right? You're missing something. So you either have granules in your cytoplasm, or you do, but we just can't see it. Make sense? With a light microscope and with staining. So let's take a look at those 2 categories. Where do they come from? Real quick, of course, in your bone marrow and your long bones, okay, you've got blood stem cells, right? And they give rise to all your blood cells. We don't care about the erythrocytes. We don't care about the platelets. We care about all these guys here. All of these guys are what? Leukocytes. These are all white blood cells. Okay, you've got basophils, neutrophils, eosinophils, monocytes, and lymphocytes. Now, most of these guys are involved in today's lecture. Innate immunity. Okay. These lymphocytes basically end up being T cells or B cells. They're really for adaptive immunity. That's chapter 16. There is one type of lymphocyte we'll mention today that's actually involved in the second line of defense. But most of your lymphocytes are going to be with adaptive immunity, chapter 16. No. Let's talk about the granulocytes. Means they have granules, as you can see, in their cytoplasm, and you put them into categories based on how they stain, and yes, you need to know this, okay? You've got basophils. It's real easy, okay? If you're a basophil, the reason why you're named that is because when we stain you with the basic dye, you're blue. So basophil, think of basic, okay? That's why it's called a basophil. So basophils, they're granule stained blue when you use a basic dye, methylene blue. Got it? All right. Eosinophils or eosinophils, okay? Easy to remember. You use an acidic dye, eosin, and their granules stained red or orange. So to help you to remember eosinophils, eosin. Use this dye, eosin, to stain them this color. Got it? Last one, neutrophils, okay, their granules stain lilac because you use 2 different colors. You use basically an acidic dye and a basic dye to stain their granules a lilac color. Now, neutral means neutralize, right? So you take an acid with the base, you make it neutral, right? That's why they're called what? Neutrophils, make sense? No. Now, both the neutrophils and the eosinophils, okay, they can phagocytize pathogens, and they're capable of diapetadesis. Now, diapetadesis just means that these white blood cells, if they're phagocytes, they leave the blood, they enter into neighboring tissues, and they're just wandering around looking for invaders. Go ahead.
Saying like pet walk.
Yeah, pet, like they're walking around, literally. So they leave the blood, they go to neighboring tissues, and they're literally wandering around looking for invaders. They can do that. Okay. Here's a quick picture of all the leukocytes, either they're granulocytes or agranulocytes. But I want you to look at their numbers here. These numbers are their relative percentages in your white blood cell counts. If you do a white blood cell count, if you're normal, right, if you're not sick, these will be the percentages of these different types of leukocytes, which means if the numbers are off, or if the numbers are high, for example, it kind of tells you you've got something going on, makes sense? It tells you you are probably sick, got it?
Do we have to know that for the test?
The numbers, no, but you should know in the next couple of slides, if you have a high number of eosinophils, what do you have, makes sense? If you have a high number of neutrophils, what do you probably have? But not the actual numbers, right? Okay. Let's talk about the agranulocytes. A meaning, okay, they do have granules, they do. You just can't see them with a light microscope, all right? That's the difference. You got 2 types, okay, lymphocytes and monocytes, okay? Lymphocytes tend to be the smaller versions, okay? Monocytes tend to be the bigger versions, that's all. Now, the lymphocytes, again, most of these end up being what, T cells and B cells. They mainly form your adaptive immunity, but there is one type of lymphocyte. It's called a natural killer cell or NK cell. Okay, they help us in the innate immunity. I'll talk about that today. Our monocytes, okay, when they leave the blood, when they mature, they become a macrophage, so big eater. So they're phagocytes. They eat big things like other cells, like your cells, for example. If your cells are infected, they can come and eat it. So they're big eaters. They eat big things like your own cells, not just bacteria, right? Bacteria are small, but they can eat infected tissue. So they're big eaters. So monocytes become macrophages, big eaters. Okay, so Edward, you're talking about what do you need to know about the numbers? Okay, this is what you need to know. So when you do an analysis of your white blood cell count, right, if the numbers are high, it can tell you what type of disease or what kind of infestation you have, for example. So if you do a whole white blood cell count, and you find that you have a high number of eosinophils, okay, either A, you've got some allergies going on, like me all the time, or B, you got a worm infection. Because you'll learn later on today that these eosinophils, they can attack worms directly. They land on them and they basically digest them. Okay? Likewise, if you have a bacterial disease, okay, you have an overall high number in your leukocytes, so all of them will be up, because leukocytes will be all of them. And you have a high number of your neutrophils. Lastly, if you have a viral infection, you will have a high number of those lymphocytes, right? High number of T cells, high number of B cells. If you have a viral infection, that you should know. Okay. Now let's talk about this process of eating other things. Phagocytosis, okay? Now most of the cells we're talking about can do that. They would be called phagocytes, for example. Now even though phagocytosis is not completely understood, it's broken down to 6 steps, and you should understand the basics of those 6 steps in terms of what is going on, okay? First you have chemotaxis, basically what? You need to go towards pathogen, right? We'll talk about that. Then you have to adhere to it, basically bind onto it. Then you need to eat it, right? Ingest it. Then you need to start to break it down, that's maturation. Then you kill it, then you get rid of it. Simple as that. Let's go into more details, all right? So this figure goes in all those steps. Everything I just mentioned, that figure talks about all of that. First is chemotaxis. Now the phagocyte is going to be moving towards the pathogen. Now what are some of the signals we're looking for? Well, the pathogen itself, right? Anything the pathogen secretes, maybe other white blood cells in the region, right? Or maybe even dead tissue or tissue that's probably inflamed, anything that's related to a disease state. Those are signals for the phagocytes to go in that direction, makes sense? And when they get there, they may likely find some bacteria, in this case, Neisseria, okay? Makes sense? That's chemotaxis. Go towards the pathogen. Find them, go towards them. Next is adherence. Literally, these pseudopodia, basically the false feet, they have to grab a hold. They have to grab the bacteria. How do you do that? It's the same mechanism we talked about in the last chapter. They're slippery. We got to basically bind onto their ligands. So there are receptors on these phagocytes that have to bind to the ligands on the bacteria. Now, of course, if there's no capsule, right? If there's no capsule on the bacteria, then it'll be an easy job, right? Find the ligand, bind onto it, right? Of course, we talked about last chapter. Some bacteria have a capsule, which means they're hiding their ligands. If they're hiding their ligands, we can't really grab a hold of them. That's why we said they're, quote, slippery, makes sense? We can't grab a hold of them. So sometimes, to help this process, we can coat bacteria with antibodies, and that gives these phages something to hold on to, makes sense? That's called opsonization, makes sense? If you coat these bacteria with proteins so that you give the phagocyte something to bind and hold on to, that's called opsonization, got it? This helps it to grab hold of something so that it can take it in, got it? Okay. Now after it actually grabs onto the bacterium, then you do what? Ingest it, literally. That's what? Endocytosis, right? It just takes it like Pac-Man, right? It just takes it right on in. Takes it in. After you take the bacteria in, okay, then you go to, let's see, the step 4 here. This is the maturization part. So let me back up. So maturation, that's step 4. So in maturation, right now you just made a phagosome. A phagosome is basically a vesicle as a result of what? This phagocyte eating that bacteria. It's called a phagosome. Okay? As I mentioned last chapter, those phagosomes do not have enzymes to do what? Break down the bacteria, right? They don't. So at this point, we need a lysosome, right, which has enzymes to fuse with the phagosome. So here we've got a lysosome, right? That lysosome has enzymes. It will fuse with that phagosome so that we can start the process of what? Breaking down that bacteria. That is maturation, got it? That's step 4. Now step 5, that's basically the killing part, right? The enzymes are now doing their job, right? The enzymes are now breaking apart the bacteria, and now in the phagolysosome. In other words, what is a phagolysosome? It's a phagosome fused with a lysosome, correct? To make a phagolysosome, right? So within the phagolysosome, that's where you actually do the killing part, which is seen here, okay? Now the last part is what? Get rid of it, elimination. Now the digestion and sometimes elimination is sometimes not complete. I mentioned earlier that your dendritic cells, which we're going to be talking about in chapter 6, when they do this, they save a little bit of the bacterium so that they can present it on the outside of the cell membrane to tell the adaptive immunity guys, hey, we got an infection going on, you may want to check this guy out, make some antibodies, get to work, makes sense? Right, for memory. So sometimes we don't get rid of everything, right? We'll save some of these parts, right? And this cell will present it on the outside to kind of get the immune system going. That's what chapter 16 got it. Now, even though, for example, our eosinophils, they can do phagocytosis. They can do that. But that's not their first line of mechanism. They can do it, but they prefer killing in a different way. So I mentioned earlier, if you've got a high number of eosinophils, you've probably got allergies or a worm infection. So what do? They literally attach to the worm. And they secrete enzymes to kind of weaken the worm, kind of break the worm down so that you kill the worm. That's their job. Let's see. This part here is kind of cool. I'm not going to hold you to it. So it's been found, for example, that these eosinophils, what they do is they literally eject out their mitochondrial DNA to attack bacteria. It's cool, but I prefer you to focus on if you have a worm infection, okay? These eosinophils bind to the worm, they secrete enzymes, they kind of make the worm soft so that the worm will eventually die. What about these natural killer cells? Now remember I said lymphocytes are basically B and T cells, primarily what? Adaptive immunity, but there's one type that we see now. These are the NK cells, natural killer cells. All right. What do they do? Well, basically what they're doing is they're looking for cells that are presenting different types of proteins on their surface. So we already know that all cells, your cells, bacterial cells, they have proteins, right, on their surface. Now, NK cells, they're going around in your body, and they're looking for some of your cells that are now presenting different proteins on the outside. So say, for example, you've got a viral infection, which means you're going to be presenting some viral proteins on the outside. Or let's say you actually have some cancer cells. If you got cancer cells, they also will do what? Present different proteins on the outside. Because they're different, right? They're different. So these NK cells look for that. And when they find a cell that has different proteins on the outside, they literally start to secrete toxins on that cell and they kill the cell. They kill the cell. That's what they call natural killer cells. They find cells either infected with the virus or maybe some cancer cells starting to come up, they just kill them. So they're natural killer cells. They're basically lymphocytes, but they're using what? Innate immunity, our second line of defense.
Do they have memories?
They don't. No memory. So memory is chapter 16. These guys here are basically, I would say, non-specific.
They're just hunters.
They're just hunters.
They don't keep memory.
They don't keep memory.
You don't belong, they attack.
They are hunters. They just find you don't belong, you're out. That's it. If you don't belong, they don't care. We're getting rid of you. So no memory here. That's chapter 16. Okay. What are these neutrophils doing? Okay, well, they produce chemicals that kill nearby invaders. Again, look for invaders, kill them. Okay, they find invaders, they secrete chemicals that kill nearby invaders. I listed them here. Again, anything you read is something I've added recently. Okay, they can make hypochlorite. That's basically in your bleach. So you've got cells in your body that basically secrete bleach to kill their invaders. They also make nitric oxide, which basically stimulates inflammation. We'll talk about that at the end of this chapter. That helps with the process in terms of bringing in new macrophages to the fight. We'll talk more about that at the end. So nitric oxide stimulates inflammation. It helps to bring in more phagocytes, more macrophages to take care of these invaders. Now, these NK guys, these neutrophils, I hate to put it this way, but they're like suicide bombers. I hate to use that term. Because they do something when they're about to die, they kill themselves, and in the process, they kill anything around them, just like a suicide bomber would do. Okay? So let's say the neutrophil is sensing it's going to lose the war. It will literally commit suicide. And when it commits suicide, okay, it's going to release everything inside of itself. So think about it. It's going to release its DNA, cytoplasm, anything on the inside is going to be released on the outside. All of that stuff becomes basically a net. It's called a neutrophil extracellular trap. It's called a net. So they basically release a net when they die. They do it on purpose. That net basically captures the invaders. It also helps to sequester some of those defenses I talked about. It helps to sequester defenses so that you eventually kill the invader. But of course, they also die in the process. So it's like your suicide bomber mission. They kill themselves, but they kill everybody around them.